Well, thank you. So I'm a full-time professor at Moran, and I am a practicing vitreoretinal surgeon. My laboratory studies angiogenesis, aberrant angiogenesis, as it reflects diseases in the macula and also in the retina. So one of the areas that we focus on is related to neovascular age-related macular degeneration. And my research interest started really from patients. I was imaging patients using infrared light when I was a junior faculty member, and recognized that there were some patients who would develop these characteristics on imaging that were associated with later development of choroidal neovascularization, which is one of the leading causes of vision loss in the wet form of macular degeneration. But not all patients develop vision loss or develop choroidal neovascularization if they had this early form of imaging. And it made me ask the question whether or not choroidal endothelial cells that are somewhat activated to migrate into Bruch's membrane or underneath the retinal pigment epithelium could be contained there, maintained there, where they may have potentially develop into physiologic vasculature. So they could perhaps provide a physiologic beneficial effect on the outer retina and not move into the neurosensory retina. Once choroidal neovascularization breaches the retinal pigment epithelial boundary and goes into the neurosensory retina, that's the usual cause of vision loss and macular degeneration. So my lab started to look at causes of choroidal endothelial cell activation, as well as what causes the RPE barrier integrity to be compromised. And so we study mechanisms of that with the goal to try to prevent either cause, but not to inhibit angiogenesis, because inhibiting angiogenesis, we believe, may lead to vision loss because of geographic atrophy and loss of necessary physiologic blood vessel growth.
I think that's a very important area. We've seen a number of papers presented of the late impact of repetitive use of VEGF inhibitors. And could you speak a little bit about that?
Yes, I mean, I think that's going to be a hard question to address, because once in the human, once choroidal neovascularization develops, we can, perhaps OCT angiography will give us more information. I'm very excited about that. But once it develops, we really don't know what the photoreceptors, what the RPE underneath it are doing while that choroidal neovascularization is underneath the photoreceptors. And then anti-VEGF can cause it to regress. But is it the anti-VEGF that then leads to the geographic atrophy, or has the geographic atrophy, the process of that, already started? So I think it's going to be difficult to assess that. But as I said, maybe with OCT angiography, but that is one of our concerns. And not only with the geographic atrophy, but the VEGF can also be important for the neural retina. You know, I also study a disease, retinopathy of prematurity, and in that I knock down VEGF in Mueller cells and find that even when I use targeted knockdown in Mueller cells to get it to a level of VEGF to a level in the retina that is the same as the control situation, or in the situation where the animal has not been affected at all, there is thinning of the outer nuclear layer where the photoreceptor nuclei are. So that suggests that VEGF may have a detrimental effect even in these cells. And other scientists have found the importance of VEGF in adult vascular beds as well as neural beds.
So it's really a very fine balancing act then.
Yes.
You don't want to inhibit too much, because you induce ischemia. The photoreceptors need blood supply. They need
exactly
source of oxygen and nutrients.
So,
and how does one try to reach that very fine balance?
So exactly. And in fact, one of the things that our lab has started to work on, based on evidence that we found, are ways to regulate the VEGF signaling pathway. So what we found is that under certain stresses that are similar to what humans experience when they develop neovascular macular degeneration, that there can be interactions of the VEGF receptors with other proteins. And these interactions lead to an overactivation of downstream signaling and angiogenesis, or can lead to loss of the barrier property. So we are looking at adapter molecules that might be involved in kind of linking up to the VEGF receptor 2 and other proteins that lead to increased signaling. And we're also looking at there's a G protein coupled receptor that we're studying, CCR3, which actually we found transactivates VEGF receptor 2 to lead to overactivation. And the idea is that if we could inhibit CCR3, which seems to be safe, as best we can tell, we have a little bit of concern where we need to do long-term studies on this, but that's one of the areas that we're going to be presenting at ARVO. But if we could inhibit CCR3, we may be able to regulate VEGF receptor 2 signaling to have physiologic and not pathologic angiogenesis.
Now, how does the interplay of immunologic factors play into this? Go ahead.
Okay, so CCR3 has historically been known to be involved in like eosinophils and allergic reactions, but it can have a role in neovascularization as well. And in fact, there is a company that's looking at a CCR3 inhibition and its effect in asthma, for example. So we don't know if that will have an effect on immunologic aspects as well. We collaborated with another lab that previously found that monocytes and macrophages were not involved in the CCR3, but I think we need more study in that area.
So do you think in future we will be doing a particular immunological screen on patients? And if so, what are we looking for? Are we looking for antibodies? Are we looking for some cofactor?
Oh, to be able to detect people who are at risk. You know, I think that at least the approach that our laboratory is taking would be we would require careful imaging. So, as I said, I'm really interested in OCT angiography and being able to potentially even detect activation through certain, if you could safely inject markers into the bloodstream of a human. And then if you find a person who seems to have the markers that would predict vision loss or risk, that would be the time that you could intervene.
Have there been any genetic variants that have been identified that are more prone to neovascularization?
So, yes, I mean there are certainly, AMD is very strongly genetically regulated with complement factor H and also HTRA and ARMS2, those gene loci, as well as other gene variants involving the complement system and many others. So in some predictive models, they can even predict the chance of developing neovascularization even in up to 90%, have been published in the literature. Whether or not somebody does better with anti-VEGF treatment or other treatments, that hasn't been shown to be necessarily. The evidence, I guess, is mixed. Maybe I should say there's controversy. But the evidence doesn't seem to say that anti-VEGF is better in certain people, who have certain genetic
perhaps
risk
a more
factors.
detailed question in that regard. Have there been specific genetic variants identified that will be people will be more prone to failure from existing anti-VEGF therapies? In other words, who's going to be a responder, who's going to be a non-responder?
I think that the evidence is we still need more evidence to be able to say that.
One of the big catchwords now, obviously, is personalized medicine. Looking at the genome and trying to identify an appropriate therapy.
Well, that's, I think, the hope, and people are still looking at that. But it's to be able to make a statement to our patients that this means this or that, and you are better with one type of management treatment. We just don't have that evidence at this point.
So tell us about the poster presentations and the other material that you will be just releasing at ARVO.
Okay, so in the presentation, we have a paper presentation in which we're looking at an adapter molecule called IQ-GAP, and that has several domains within the protein, including a RAC1 binding domain. And we're interested in RAC1 because when choroidal endothelial cells are activated, RAC1 is activated. It's a GTPase. So it's like a biologic switch. It is activated by certain guanine nucleotide exchange factors and inactivated by GTPases. And when RAC1 is activated, the endothelial cells start to migrate. So we've found that IQ-GAP can be an adapter molecule that may be bridging between the CCR3 G protein coupled receptor that I was discussing and VEGF receptor 2. And when we knock down IQ-GAP, we can inhibit CCR3 or choroidal endothelial cell activation and migration. So that's one of our presentations. And then our poster is really looking more in the animal model where we use a CCR3 inhibitor. So what we found is that in humans, so this is a previous studies, that CCR3, this protein-coupled receptor, is increased in expression in choroidal endothelium and in human donor eyes, and it's increased with age. It's also increased by stresses that are similar to what people with macular degeneration experience, like reactive oxygen. And in our poster, we wanted to see, well, first of all, if we could use the wild-type mouse as a way to study CCR3. And indeed, we found that CCR3 expression was increased with age, and we also found that it was activated with laser, which we know from previous studies to increase reactive oxygen. So one of the concerns was if we inhibit CCR3 to reduce choroidal neovascularization, and it's expressed in ganglion cells and other neural cells in the retina, could it potentially injure the retina by inhibiting it? And so we set aside to study that, and what we found was that the CCR3 inhibitor at a dose that reduced choroidal neovascularization significantly caused cell death within the choroidal neovascular lesion, and it also caused cell death in some cells overlying the retina that we have not yet been able to identify. So these might be cells that they may have changed their markers because they were starting to undergo cell death, or they may be cells that we just haven't found the markers yet for. So they could be, we looked for microglia. That was what I was thinking they might be, or endothelial cells, but they didn't co-label with those. But that suggests that there might be some concern that their long-term safety. We've studied out to 7 days in the model, and there's no change in the retinal thickness. There's no change in apoptosis determined by activated cleaved caspase 3. So other parameters seem to be safe, to indicate that it's safe. So we're hopeful, but I think we still need to study a little further.
Let's change to the issue of translational implications from the lab to the clinic. Which of the areas that you're currently working on has the shortest timeline to translation?
So I think the CCR3 inhibitor. There actually is a human CCR3 inhibitor that they're studying for asthma. And we wanted to design a study to test it in humans with AMD. And I think right now, both from the company and it's a funding issue. So they're really working on the asthma side of it. And of course, to do a study like that in humans would require some time because the standard of care right now for treating neovascular AMD would be anti-VEGF. So it may be a situation where can you reduce the number of injections that are needed by looking at many of us we'll look at the optical coherence tomogram, the OCT, for guidance about whether or not to give an additional injection of anti-VEGF. So we may be able to extend the period of time in between injections in patients with the CCR3 inhibitor.
Well, that raises another issue, alternative routes of delivery for any of the anti-VEGF agents. And there's obviously work going on in this field. What do you think is the most promising of those?
If I can go back to the CCR3 inhibitor, the thing that's nice about that is it's oral. Right. And it has been tested in healthy, I think mostly men, I think was what the study, and it has been, at least has received approval for the asthma part of it. So it seems to be healthy. But we don't know if that is bioavailable in the retina, so that would be part of the issue. So as far as alternative dose, oh my gosh, there's so many different possibilities. So it does seem that in neovascular AMD that monthly injections is very important, but from the clinical trials, and that when you treat and extend, patients still can maintain good vision over a period of time, and there's some variability in individual patients, so at least what I find in my own practice. So the concern, I think, is that if you gave something that always inhibited VEGF, that it may have a detrimental effect if the natural course of the disease in that individual patient was to have VEGF go up and then go down, right? So I worry about things that would be forever. However, the slow release formulations that might inhibit VEGF signaling for extended periods of time, similar to like the Ozurdex and some of the steroid formulations, that would allow patients not to have repeated injections, I think would be very good. That would be a very good approach.
Switching now to messages for patients. What is the promise and potential for patients in the clinic of the work that you've been doing?
Well, I've been focusing on neovascular AMD, but with a second, or not a secondary, but with an additional focus of trying to reduce the formation of geographic atrophy or to do it safely. And so I think the hope would be to be able to have other treatments that help to reduce the burden of injections from anti-VEGF or even alternative treatments, and to be able to have better quality visual acuity. I mean, many times we talk about legal blindness. Anti-VEGF reduces legal blindness, but there's still other aspects that patients with macular degeneration suffer from, like reduced contrast sensitivity and a number of other distortion. So the hope would be to be able to have a very safe, less burdensome and drug that could even catch very early forms of macular degeneration and keep them at bay, and potentially even without the need of injections after a period of time.
Are you currently recruiting any patients for clinical trials?
So I'm not. I was, as I said, we had hopes to have a clinical trial with the CCR3 inhibitor, but it does take a period of time, I think, to be able to safely design a clinical trial to even get a sense of efficacy, and we just are looking for funding, I guess, or sorting that out from funding. Can I say something else? Because you can edit this, right?
Go ahead.
Yeah. I mean, we presented this to the company. I don't know if you want this, but we presented it to the company, and the company wanted us to come up with something to do in a month. There's just no way we could do that, because you have to be able to, the standard of care is arguably 3 monthly injections of anti-VEGF, and then you could treat and extend. So anyway, that's the reason. I don't know if there's a different way you want me to say that.
If all goes well with the CCR3 inhibitors, what do you think the timeline is for putting a FDA-approved therapy on the table?
Gosh, I
know that's always a difficult question.
I don't know. I think I would like to see whether or not it helps to reduce the burden of injections first.
Right.
And then that if the drug is approved, I think, or it's close to being approved for asthma. So it may even be, a different indication potentially. So I don't know.
It's not possible to predict at this point.
It's not possible. It's not possible to predict at this point.
So I mean, with some drugs, you can see the progression of the clinical trials, and you can get a sense of the horizon. But you say it's a little too early at this point.
I believe, I think it's too early. But I see it as a possibly very safe drug.
But the initial studies are very promising.
Mm-hmm. Yes.
Based on safety and efficacy.
Yes, and also we understand the mechanisms better. So I think that's also helpful to know, to even predict where we should look if there are safety issues that arise in the future that would be hard to know from the studies, the short-term studies. I think knowing the mechanisms gives us an idea of what we need to be looking for as possibilities or preventing.
Right. Turning to your laboratory and your resources and your needs going forward, could you tell us about that?
So my laboratory is NIH funded, so I have several R01s. I'm very thankful to the National Eye Institute and NIH for funding to be able to do these investigator-initiated questions, which I think are so important. And I also have funding from the March of Dimes that helps to support some of the studies we do on erythropoietin in retinopathy of prematurity and also in the neural retina. So, and we also have funding from the Mildred and Carl Reeves Foundation, where we're gathering donor eyes from humans with macular degeneration to look at genetic variants. And we have funding from Research to Prevent Blindness. As a department, I don't have individual funding, but RPB helps to support our department, and that's important. And then one of my the research assistant professor in my laboratory has just been funded from the Knights Templar Eye Foundation to study retinopathy of prematurity and some of the neural central aspects of that. And then Knights Templar is a really wonderful foundation who has also provided a number of travel awards to ARVO this year.
Excellent. Is there anything else that you'd like to tell us about?
I don't believe so. Thank you very much.
Yes, thank you. It's been wonderful and comprehensive and very insightful. We wish you the very best of luck, and I promise you the support of everyone out here.
Thank you very much.
Okay.
It's been a pleasure.
Thank you. Professor Hartnett, tell us a little bit more about CCR3.
So one of the exciting things about CCR3 is it doesn't appear to have a role in any physiologic events in the body. So it's historically been known to be activated in allergies and involved in eosinophil activation. So it's being looked at in asthma. It also has a role in neovascularization, and mainly in pathologic neovascularization. So what this offers is that if you inhibit CCR3, and if it has a beneficial role in reducing choroidal neovascularization in macular degeneration, or in reducing the number of injections of other agents, such as anti-VEGF agents, that a patient needs to go through, that this could be a safe and very effective way of treating neovascular AMD, and potentially, as we learn more about its efficacy, even in preventing choroidal neovascularization or treating very early forms to prevent vision loss from it.
That's very exciting, particularly the preventative aspect.
Yes. I mean, when I say prevention, I think it could be preventing. It may prevent what we're seeing is that it plays a role in the activation of choroidal endothelial cells from the choriocapillaris to migrate toward the retinal pigment epithelium and then into the neurosensory retina, where they can then grow to develop choroidal neovascularization, which is what causes blindness in neovascular AMD.
So you could potentially identify people via genomic studies at risk and start them on preventative CCR3 inhibitor therapy.
So potentially at some point, I think what I would do first is also image them to see whether or not they actually had any evidence of abnormal either drusen or other features that we now recognize as associated with neovascular age-related macular degeneration. So people may be at risk because of genetics, but not everyone who has a genetic variant actually develops neovascular AMD. So I mean, there's no reason a person should, even if it's safe, there's no reason a person should take a medication if it's not likely to be of help.
So this would be an imaging-directed preventative therapy.
Yes, I think so, but never, with future, I think we're going to be using genetic testing as well, as we know more. Yes.
Wonderful tools for a bright new future.
Yes, I think so. It would be very exciting.
Excellent. Thank you.
Thank you.